Hi, I'm Jonah Comstock, Editor-in-Chief of Farm Forum. I'm here at CAR TCR in Boston. I'm with Jason Noon of Nona Biosciences. And Jason, what's your role there?
I'm a principal scientist with Nona Biosciences, kind of a group leader in the research team.
Great.
Yep.
Well, thanks for chatting with me. Tell me a little bit about Nona Biosciences and what you guys are here at the show to talk about.
Yeah, so Nona Biosciences is a kind of a technology-driven service company. We were spun out of a bigger company called Harbor Biomed in the middle of 2022. So we have a technology platform called a fully human heavy chain only antibody mice. So it's a transgenic mouse platform.
Oh, interesting.
We were really the world's first to develop these mice that can make fully human heavy chain only antibodies. Um, and these HCABs, this has been validated in the clinic as well, because our parent company, Harbor Biomed, developed numerous HCAB-based therapeutics, whether bispecifics, T-cell engagers, many of which are already in the clinic and have shown efficacy and good safety. So what Nona is doing now is we are extending this HCAB platform to CAR T and other cell therapies because we see a huge advantage in this
Yeah.
platform.
So potentially you can get what you need to do CAR T out of a mouse instead of out of a human donor?
Yeah, so with CAR T, right, and other CAR therapies, what's expressed on the surface and exposed outside of the cell is the antigen binding domain, and that consists of an antibody, right?
Right.
And traditionally, that was from antibodies that were generated in like wild type mice, okay, and those were converted to scFvs. So that would be one heavy chain and one light chain linked together by a small peptide. But those have been found to be immunogenic in patients, and that has been widely published, and that's known to affect the persistence of CAR T cells. As when they're administered to patients, there will be an immune response, and that will
Right.
be generated against that antigen binding domain. So other ways of improving that has been to either switch to like a camelid VHH, which is smaller, and it doesn't have a linker, and it also doesn't have a light chain. So being a much smaller sequence, it has a lower risk of immunogenicity, as there's less epitopes available in the sequence to be immunogenic. So that's one way. But what we have done is we also have this uh, from our platform. It makes this very small, heavy chain only antibody, but it is fully human, as we have cloned the human VH genes into the mice.
Mm. And so
Okay.
that makes
Yeah.
it more resistant also to
Yeah. So
immunogenicity.
thereby all when we use this HCAB, the VH for CAR T cell development, all of the antigen binding domains on the surface are going to be fully human and have lower risk of immunogenicity, and potentially enhancing persistence as well.
So,
Yeah.
what's the data you guys brought today? You had a poster?
Yeah. So yes, we have a poster for this conference. That is on. So in the past year that Nona has existed, right? We have been working on extending or extending our HCAB to cell therapy, and what we've been doing is we developed a rapid screening platform to identify those HCABs, those heavy chain only antibodies that are ideal for CAR T cell therapy. And what we did is we developed a direct functional screening platform, right? And that's basically reporter cell, a CAR T reporter cell, that we can build a library of HCAB cars in a library and screen them in this CAR T reporter cell rapidly by stimulating those cells with cells that express the target antigen, and we can then sort out the GFP positive reporter cells, and then we can do rapid next generation sequencing to identify all of those HCAB clones that strongly activated those reporter CAR T cells. And what we can do is we can provide this as a service to any client and help them develop their HCAb-based CAR T therapies, and we can do this very rapidly.
Interesting.
Yeah, and we also, we are so today, or from at this conference, what we were presenting on is we were presenting on both the extension of our HCAb2 CAR T, and also our development of this direct direct CAR-based HCAb discovery platform that we've developed.
What are some of the trends you're tracking at the show? What have you learned? What are you sort of thinking about in terms of like where this space is going next?
Yeah, so that definitely relates to kind of Nona's niche in cell therapy, and I see many talks talking about persistence, right? How persistence is such an issue, and there are so many ways of enhancing persistence, and that can be focusing on the state of the cells themselves as they are isolated from patients. That can be the manufacturing process of the cells, but there isn't much. There's not much focus on what about the actual antigen binding domains that are exposed on the surface of the cells. And what I'm seeing is the trend is that so many companies are still using the traditional antigen binding domains, which consist mostly of these SCFVs. All right, and those can be humanized SCFVs, which originally come from mice. So these are still going to be have really high risk of being immunogenic, especially still having a mouse sequence and having an immunogenic linker. And so many companies are still using that. And so I think it's such a great opportunity for Nona Biosciences to be able to connect with all of the companies in the CAR T space and share what we have to offer them to potentially improve their next gen CAR T therapies.
Yeah,
right.
That's really interesting. It makes a lot of sense. So,
Yeah.
so whatever innovative thing they're doing that's improving the chances of the therapy working, it's going to be hindered if this part of it is suboptimal, right? Like,
yeah, this is always going to limit whatever increase in persistence that they will want, and in some cases you can see in the literature that the immunogenicity to the antigen binding domain has really killed their therapy, and connected to strong antibody responses to the binding domain on the surface of the CAR T cells, or even T cell responses, right? And if the binding domain is immunogenic, T cells in the patient can eventually also kill your CAR T cells. So yeah.
So really, you want to get the word out for people to know maybe that this exists and that there's an opportunity to improve on there.
Yes, exactly. I think it's a huge opportunity, not just for us, but for any company that's looking to develop new binding domains for their CAR T therapy. If they have a new antibody discovery campaign they want to do to identify new binders, I think it's a perfect opportunity to use Nona Biosciences and our platform to develop these fully human HCAb-based CAR T cell therapies. Right? And this can be extended not just to CAR T cell, but also other engineered cell therapies.
Any final thoughts?
No, I mean I think it's been a great conference. We've had so many amazing presentations and been able to connect with a lot of great companies. So it's been great. Thank you.
Thank you so much for speaking to me. It's been great, Jason.
Okay. Thank you.